Everyone, in this video we will be discussing about the Sanger sequencing. So what do you mean by a sequencing? The first thing we have to know is what is a sequencing. When we discuss about the sequencing, there are various methods of sequencing like Sanger sequencing, Pyro sequencing, Maxim-Gilbert sequencing. So what we do by this sequencing? Suppose we are having a piece of DNA and we want to know how the nucleotides are being arranged. That means how the A, T, G, C, which are the nucleotide bases, how they are arranged and they are forming a DNA. So for knowing that, we will use a sequencing method. The Sanger sequencing, it is also known as a chain termination method, and we are using the Sanger sequencing to sequence the base pairs of 900 to 1000 base pairs. So we have to know what are the reagents required for the sequencing to be done. So for the sequencing, the first thing which we require is a DNA template, and we must know that the reagents which are required are the same as we perform a PCR reaction. What happens in a PCR reaction? We need a template DNA, we need ATP or the DNTPs. It may be ATP, GTP, CTP and TTP, as well as the primers and the DNA polymerase enzyme. Additionally, in the sequencing, we require DDATP, DDTTP, DDGTP, DDCTP, or in general, we can tell that it is DDNTPs. Okay, so what do you mean by that? What is the difference between the DNTPs and DDNTPs? Let us see with this diagram and see the structure of a nucleotide. In this nucleotide, it is having a 2' group and a 3' group. And this is a DNA. This is the structure of a DNA. In case of a DNA, in the 3' group, it is forming a OH group. And in case of a 2' group, it does not have a OH group. Only the hydrogen group is present. And in the 5' end, it is having one phosphate group. And here you can see a nitrogenous base is present. This nitrogenous base can be anything like adenine, cytosine, thymine or guanine. Let us see the structure of the DNTPase. NTPase means nucleotide triphosphate. This phosphate group which was present in the 5' and in case of a DNA, they are in alpha, beta and gamma form. This is the alpha, beta and gamma form of the phosphate which is attached near the 5' and it is the alpha phosphate. Next to is the beta phosphate. Next to it is the gamma phosphate. Here there is a OH group which is present and this OH group is responsible for forming a phosphodiester bond or it is responsible for attacking the alpha phosphate. Suppose we are having OH group over here in the 3' end and this OH group will attack this alpha phosphate of the upcoming nucleotide and will form a phosphodiester bond if it is having the upcoming nucleotide is in the DNTP form. But whenever it is having DDNTPs, it cannot form any bond. Why? Because in the DDNTPs, you cannot find a OH group present in the 3' end. It is known as DDNTPs. Why it is known as DDNTPs? Because in the 3' OH 3' end it does not have a OH group as well as in the 2' and also it does not have a OH group. That is the reason it is known as dideoxynucleotides. Okay, this was a major difference between the DNTPs as well as the DDNTPs. What is present in the DNTPs? OH group is present in the 3' end. But in case of DDNTPs, no OH group is present in the 3' end. Only the hydrogen group is present. That is the reason it cannot form a phosphodiester bond with the upcoming nucleotide. Let us see with this diagram how this is happening. Suppose we are having a template DNA. This is the template. This is the template DNA, and whenever there is a phosphate or whenever the DNTPs are coming, it is forming a phosphodiester bond. But whenever DDNTP is present, the upcoming nucleotide cannot form a phosphodiester bond. So we are clear with what are the reagents required. Next we will see how the process is taking place. So whenever someone is giving us a DNA sequence and they are telling for performing a Sanger sequencing. So when they will give us the sample are in the double-stranded form. We can see here the genomic DNA which is present, they are in the double-stranded form. Okay, the first thing which we have to do is if the concentration or the amount of DNA is sufficient, then we can perform the sequence directly, or else we have to do a PCR PCR reaction for amplifying. We can do a PCR reaction for amplifying. Okay. So this DNA which is present in the double stranded form, first of all, they must be changed into a single stranded form by the process of denaturation. The other thing which we require is a primer. How can we design a primer if we don't know the sequence of this DNA? So suppose this is a DNA, stretch of DNA, and we have to design a primer. So in the end, what we have to do is we have to ligate some of the known product. If we are ligating some of the known product, so here by seeing this known thing, by seeing the known sequence, we can design a primer. So already we have designed a primer. So what we have to do now? We have to extend this DNA. So here the primer is NL. First of all, we have the denatured DNA. This is the denatured DNA. And the things which we required was same as a PCR reaction. We require a primer. We require a primer. We require a DNTP. These are the DNTPs and the DDNTPs. The DDNTPs, they are fluorescently tagged. And we require a DNA polymerase for extending this primer. So let us see the second step. What is done? So the DNTPs are incorporated here, and they are forming a phosphodiester bond with the help of this DNA polymerase. This is being extended till how much it will extend itself until it get a DDNTPs. Until it get a DDNTPs, the chain will be elongated. Whenever the DDNTPs is being incorporated, it will stop the elongation and the chain termination will take place along with this fluorescently tagged DDNTPs. The next thing which is exactly done. Suppose we are having a stretch of DNA and we want to know the sequence of the DNA. We have to first of all do a PCR kind of reaction. That is 4 PCR reaction will be done. Along with it, in each tube there will be DDNTPs added. Here you can see that in this first tube the DDTTPs are added. That means dideoxythymine triphosphate. Here in the second tube, DDATPs are being attached, that is dideoxyadenine triphosphate. In the third tube, DDGTP, that means dideoxyguanine triphosphate. In the fourth tube, DDGTP is being attached, that is dideoxycytosine triphosphate. We can see here a DNA sequence. And the sequence which is formed is a complementary strand. Okay, we are having a sequence of a DNA. This is the sequence of a DNA which needs to be sequenced. Okay, so we are giving a DNA template. The DNA template which is being used in the reaction is in this form. Okay, in the first tube, what we saw that we are performing a PCR reaction and we are dividing it into 4 tubes. And in the first tube, we are adding ATP, DD, ATP. In the second tube, DD, GTP. In the third tube, DD, CTP. In the fourth tube, DD, TTP. This is being extended till it is getting the DNTPs. Whenever it is getting the DDNTPs, during that time it is being terminated or getting a different stretch of DNA. That means they are of small to big size. So accordingly, in the gel electrophoresis, it will be arranged. We can see here the sequence we are getting, the sequence which we are getting in this gel is from the 3' to 5' direction. Okay, so in the base, what we got is we got T. So here it is C. The next thing which we got in this gel is C. So here is C. The next is A. Here it is A. Here we can see it is A. The next is G. We can also see here the G. In this way, it is going from 3' to 5' direction. As we have discussed that this strand which we are getting in this gel, it is not the original sequence of the DNA. It is a complementary sequence. So here you can see that this is in the T. Okay, let us now write the sequence of the DNA. It was A, G, T, C, A, T, T, A, C, G, G and T. And the sequence, this is in the 5' to 3' direction. 5' to 3' direction. So when we do a complement sequence, here it will be 3' and so what we got here? We got T, C, A, G, T, A, A, T, G, C, C and A, and it is 5'. So this is the sequence of this template. So when we do a complementary, we can get the original one. This is the original one. And what we got is, this is the sequence. This is the sequence which we got from gel. If we complement this sequence, complement this sequence to get the original one, to get the original DNA sequence. This is how the sequencing is being done. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.